and our director is Dr. Peter Garino. Now, SHARP, if you've worked on any of the HIV prevention research networks, you'll know that SHARP is also part of the Statistical and Data Management Center for HVTN and MTN, as well as HPTN. The way SHARP does most of the work is that we're a very team-oriented environment, and each protocol is assigned a study team that will work at SHARP on the study. The team consists of a clinical data manager, statisticians, clinical programmers, lab data operations programmers, and data managers, and also clinical safety associates. The teams are supported, as in most organizations, by other groups. We have admin, finance. We have people who do MEDRA coding, which is coding of adverse experience data so that it may be easily analyzed, data management, lab programmers, and then also quality systems, the people who make sure that we have all of our SOPs and follow them. And of course, we're, as you might expect, for a statistical and data management center, we have a large IT department. The SDMC consists not only of SHARP, but also another component is our faculty statisticians. So I mentioned Deborah. She is not only our principal investigator, but she is also a faculty statistician, and she is the lead statistician on the HPTN 083 study. We have a number of other statisticians, PhD level statisticians. So there's Tom Fleming, who in the past had been the PI of the HPTN Statistical and Data Management Center. Also Jim Hughes, who is the protocol statistician for HPTN 084 and 078. And he is also the person who is generally chairs the study monitoring committee for the HPTN. We also have Brett Hadscomb, who is the HPTN 074 statistician and also works with Deborah on HPTN 083. And we have a couple of newer faculty, Tim Scallon and Sahar Zajana, who will be working on some of the newer studies coming up. I think it's probably less well known that the HPTN Statistical and Data Management Center also has a math modeling center. The HPTN Math Modeling Center is located not only at Fred Hutch with us, but also we have a group at the Imperial College in London. These folks then perform a number of math modeling, a lot of work for us, and I'll talk a little bit about that in shortly. As mentioned, the HPTN Statistical Center and SHARP is located in Seattle, Washington at the Fred Hutchinson Cancer Research Center. So what does the Statistical and Data Management Center do? We have a variety of roles that we perform for the HPTN. Our faculty statisticians provide statistical and study design expertise for the network. We design, along with the protocol team, the case report forms that are used to collect study data, and also program and work with the protocol team on designing computer-assisted self-interview questionnaires, which are usually known as CASI. We also conduct study-specific data management training for the clinical research sites. An example of this is coming up for HPTN 084, where our clinical data manager for that study will be working with the statistical data management centers who will be on, sorry, not statistical data management centers, the research sites who will be working on the study. We also provide support to the site in matters related to data management and data transfer. We provide centralized data management and quality control of study data. We then also integrate other data streams besides the case report form data, and that includes lab data and other data that comes into the Stat Center. And then we also then provide support to the HPTN Laboratory Center for a number of things, including specimen shipping lists and support of the LDMS laboratory data management system. We also maintain, distribute, and archive study data when the studies are complete. We conduct clinical review of the safety data to make sure that it is complete and makes sense. We, as I mentioned before, we code adverse experience data so that it can be easily analyzed. We also report on study progress, so those are the enrollment reports and retention reports and other reports that we produce throughout the life of a study. We also report on the clinical research site performance, and this includes data management quality at the clinical research site, accrual, and whether they're meeting the protocol targets for enrollment, and also are they retaining participants as per protocol. We produce a number of specialized study reports, including reports for the Study Monitoring Committee and also for data safety and management, data safety and monitoring boards. We also provide centralized statistical analysis for presentations and publications. As I mentioned, we have a math modeling group. This group designs and conducts mathematical modeling and simulation studies to support HPTN research activities. Among the things that they do are assess population-level impact of the interventions that we're testing to see how they may suppress the HIV epidemic. Their work also informs the design and the conduct and the final analysis of our studies, and they also conduct methodological and validation work. They do work for us before the study starts and help inform protocol development. They also inform our questionnaires so that they receive the data that they'll need to be able to do their math modeling analysis during the study. They also model study objectives to see if we can actually meet them per protocol. And as I mentioned, they contribute also to survey instruments. During the study, they take interim data and do analysis to see if the intervention impact is what we expect it might be at the population level. And then when the study is complete, they review study data to, again, assess what the impact of the intervention that had been tested might be at a populational level in the countries included. And so you can see here that they've worked on before and during the study on HPTN 078 and also HPTN 082 and the AMP studies. And so their work is quite important. Okay, now I would like to speak a little bit about data management at the Statistical and Data Management Center. So a big part of the data that we receive is case report form data. And in the past and also currently, a lot of the data was collected by site staff entering the data on a actual paper form and then transmitting that information to us. We are in the middle of switching over to electronic data capture, and I'll speak a little bit about that system as well. So the system that we're switching to is called Medidata Rave. It's an industry-leading electronic data capture system and data management platform for the capture, management, and reporting of clinical, operational, and safety data for HPTN studies. It's unlike DataFax, our former system. It is online, and it's a web-based interface, and site data management staff enter the data online rather than faxing it or transmitting it to us as they did with DataFax. It's recognized as a leader in electronic data capture products. The company Medidata has been in business since 1999, and it currently supports over 400 clients. 9,000 trials have used Medidata Rave to collect data, and that includes over 200,000 research sites. So it's quite a huge endeavor to switch over from one data management system to another. So why would we want to do that? I mean, we are currently having to redo all of our data flows and all of our procedures, and it's quite a large endeavor. But part of the reason that we are doing this is that other networks are doing it as well. And it is also endorsed by DAIDS, and it is also a product that makes it easy for the monitors to go to the sites and log in and review data, and then also sign off on that data so that there's an electronic record that a monitor has looked at the data. And this product has a proven track record with a number of our partner organizations, including Gilead, who provides drug for a number of HPTN studies, Janssen, and also Johnson & Johnson. So why would we want to start using an electronic data capture system rather than the system we had been using? I think a big part of it is that site staff really like the real-time data and consistency checks rather than faxing in data and finding out 2 weeks later that there was missing data or an error on the data in a QC report, as we used to send out. They get immediate feedback if a particular data item is entered and it doesn't seem correct, or if there is an item that's missing. We don't need to worry about illegible handwriting when data is entered directly into an online system. It performs a number of calculations automatically, such as we hope that in HPTN 084 it'll automatically check whether the grading of abnormal lab values is correct or incorrect. And also it just provides better data management quality performance for the sites and for us. We think it's a really user-friendly product, and also some of the other reasons to use it are that it'll provide much more real-time data access for reporting. This is especially important for safety reporting, so that we people reviewing the safety reports have very much up-to-date safety data. I am going to move on here. Okay, and in addition to data on case report forms or electronic case report forms, we often collect behavioral data using computer-assisted self-interview, CASI, or some of our questionnaires actually have an audio component where the question is read to the participant, and those are called ACASI for audio computer-assisted self-interview. There is also other kinds of data that are submitted to SHARP. As I mentioned before, there's laboratory data that is submitted to us by the laboratory center and other laboratories. There is also data from devices such as the WISE pill device that was used at HPTN 067 and 069. And then also some of our studies are using SMS text messages to support the study, and we also receive those data too. I'm going to move on and talk about reporting and analysis done at the Statistical and Data Management Center. Some of the reports that we produce routinely for HPTN studies include accrual and retention reports so that we can monitor study progress and make sure that participants are being accrued at the rate that we expected, and also that participants are being retained throughout the study. We also very importantly produce safety reports that are reviewed by the study monitoring committee or the data and safety monitoring boards or our independent safety reviewers to make sure that our participants in our studies are participating safely, that there's no adverse experiences, and that nobody is being harmed by participating in the study. We also produce along with the other components of the network, we produce the clinical research site performance and data management quality reports that report on, again, accrual, retention, and how well the site is doing their data management. We produce, as I mentioned, reports for study monitoring committee and for the data safety and monitoring boards. And as the study progresses, and especially as the study ends, we work with the protocol team to do the analysis needed for publications and also for presentations at important conferences. A large part of our work is also supporting the HPTN scholars and helping them with analysis for their publications and presentations. When the study is completed and most of the analysis has been done on the study, we also then produce data sets that are posted on our Atlas web portal, and that then provides additional access for HPTN investigators and other investigators long after the study is ended, so that they can continue to use the data and benefit from it. Okay, and now I'd like to just talk a little bit about some resources for people out at the research sites. If you'd like to know more about the Statistical and Data Management Center and SHARP, SHARP has a nice website now, and it's just www.scharp.org. And you can learn more about what we do. As I mentioned, there's also the Atlas web portal. It's just a website that we use to post reports, documents, and data sets for various SHARP projects and HPTN studies among them. And the address for that is easy to remember. It's just atlas.sharp.org. When you go to Atlas for the HPTN, most of the information, therefore, about HPTN studies does not require an account on Atlas. You can just go to Atlas and see quite a bit of information about the studies. This is what the page here is, what it looks like. It lists all of the HPTN studies first on one page. And then each study has its own set of pages on Atlas. And on those pages, there are reports posted, as I mentioned, accrual reports and retention reports and other various reports needed for the study. And also there's documentation for the research sites. And for example, on this is the HPTN 083 Atlas page, and it has Medidata Rave training materials posted there, and it also has the paper CRFs and other documents that the site may need for HPTN 083. And as I mentioned, we also, when the study is complete and most of the analysis has been completed, we post data on Atlas so that investigators have access to it. I want to mention a sometimes overlooked resource, which is the HPTN Manual of Operations. Chief among those things that are related to the SDMC work is the publications policy, which guides HPTN investigators in publishing study data. And also Section 12, the study implementation section, discusses, among other things, reporting and also release of data from the Statistical and Data Management Center. These are my acknowledgments. And I'm not sure if we're taking questions at the end or now, but I'm happy to answer any questions.
We just have a couple of questions while Phil sets up. The first one, Dr. Emmel, is have you evaluated any other free access EDC such as REDCap?
We did go through a process to select the system that we are using, and we had a number of candidates that we reviewed, and we felt like we selected Rave. Among other reasons are that it was being evaluated by other networks, and it ended up being selected by other networks. And we also felt that it is being used in a number by pharmaceutical companies, and it does seem to be an industry leader. So that's why. I mean, we do for the CASI questionnaires, we do periodically do a review of software. We know that new software comes out all of the time, and anyway, we do periodically evaluate these things, but with Rave, it seems like it's becoming a DAIDS standard, and so for the foreseeable future, anyway, we'll be using Rave for EDC.
Thank you, Dr. Emmel. There are a couple of other questions, but I'm going to hold those to the end so that we can make sure that Philip has enough time to present. So with that, I'm going to turn it over to Philip Sullivan.
Thank you. Thank you, everybody, for gathering today and joining the webinar. Thank you, Linda and Jonathan, for your introductions and presentation. I'm going to do my best to be brief and give an overview of the HPTN Laboratory Center. So I have no disclosures to make. And today the overview will cover the LC structure, our organization, our goals, staff activities, support, and we'll introduce some network evaluation criteria that is new within the past year. For anybody who is not familiar with the U.S. or Hopkins, we are located in Baltimore, Maryland, and that's shown here by the red pin. And with the exception of Dr. Anderson's laboratory in Aurora, Colorado, all of the LC activities are based here. The LC is headed by Dr. Eshelman and the recently appointed Dr. Mrzinski, and Estelle is our deputy director. We have 15 investigators and over 50 support staff, and this is across 6 CLIA certified labs and 9 state-of-the-art HIV research laboratories. So the LC is shown here in the center. It is made up of 5 different components. We have an administrative core, a quality assurance quality control core, and laboratory cores, and support labs, and last, consultants. The virology core is further divided into HIV diagnostics, HIV incidence, molecular analysis, and phylogenetics. In the pharmacology core is divided up into the pharmacology core, pharmacology support lab, run by Dr. Anderson, as mentioned, in Colorado. The clinical chemistry lab, where we do some ARV screening, and the pharmacogenomics lab. The STD, immunology, and toxicology labs are also on the same level. The QA/QC core, which I am a part of, is comprised of protocol specialists, Vanessa, Denny, Paul, and myself. We have a regulatory compliance officer, Barbara, and we maintain a CLIA lab here. The sample testing and management team is Stephanie, Tammy, and Michelle, and our international coordinators, who are also protocol specialists, are Yao, Danelle, and Liba. The HPTN LC has 3 goals: to provide leadership expertise and laboratory support to all of the HPTN, to support our site labs and ensure the quality of all the HPTN lab results, and to advance HIV prevention science with assay development and our biomedical research. Apologies, some slides are mixed in that were supposed to be removed. The Hopkins-based QA/QC team is pictured here. The 3 individuals on the bottom are Liba on the left, Yao on the right, and Danelle at the bottom. Yao and Liba are stationed in Africa, and Danelle is stationed in South America, and they help with regional sites and laboratories in those areas. All of our protocol team members help with development, study-specific procedure manuals, HIV testing algorithm development, and protocol training and site training as needed. All of our coordinators are certified laboratory scientists, and we cover a diverse range of laboratory fields. And the LC has prevention trials participation and guidance dating back to previously named organizations before it was called DAIDS. And we work with U.S. and non-U.S. CTU and CRSs. For anybody unfamiliar, HPTN operates all over the world, and there are some markers here denoting some active sites currently in almost all continents. The Laboratory Center's network-based activities involve performing research to support the network agenda. We help guide protocol objectives and development. We evaluate HIV/AIDS related testing regarding diagnosis and treatment monitoring. And our staff also participate across networks whenever possible to help harmonize the laboratory related activities. The LC will work with sites before, during, and after protocol starts. Before a protocol is initiated, we can visit sites to help review workflows so that the impact of a study can be assessed on the laboratory. We'll assist with completing protocol analyte lists, and these are the sometimes large and cumbersome spreadsheets that are distributed. These contain all of the testing that's associated with a protocol at a particular site. We will also work with sites to ensure that they have proper quality assurance and proficiency testing programs in place as they relate to protocols and PALs. And we work with sites to ensure necessary staff are trained to use LDMS, and this is done through our partners at Frontier Science. We also assess staff for their completion of the GCLP training requirements, local safety standards, and IATA shipping standards. The Laboratory Data Management System is a DAIDS mandated system for the management of study-related samples. The system is to be used for all samples being sent to the LC for testing or any samples being held at the site for future testing. Chains of custody are protocol-specific documents that sites produce and the LC reviews for all of the studies involved in HPTN, and they detail a site's sample handling scheme and their related procedures. Here we have two screenshots as examples from LC training materials that can be provided on sample management for sites. Sample collection and handling are important parts of all chains of custody. Many different samples are required across protocols, and each sample has different means of collection or handling. So we always remind staff to review sites' detailed order of draw for blood collections and review associated manufacturer recommendations for supplies. And this is just an example of a manufacturer-supplied material for order of draw. It also notes a proper mixing technique for inversion. We stress the sample collections and sample handling in study documents because they have some serious repercussions further on with study samples. It's important to know what each protocol requires, how samples are being provided to the laboratory and the storage facilities, and that they have all been appropriately treated up to each point of exchange or storage. The LC does its best to provide simple guidelines for sample processing so that each site can treat all samples similarly while still following their local procedures. The more uniform all samples are treated, the better our study results can be. And networks will vary in their direction of sample handling and collections, and it's important to know where HPTN differences are so that they can be addressed before any issues occur. This is an example of a tube that is labeled with a Sharp provided primary container label, and it still has the manufacturer information visible so that the integrity of the sample can be verified when it's being processed. And chain of custody designated documents, like the one shown here, are used for accompanying samples to make sure that data from the visit and collection and regarding the sample can be transferred accurately into LDMS, and that any errors that may come up later can be resolved by checking the documents against the LDMS entries. Again, each protocol has different requirements which are detailed at various levels through study documents. Sample completeness is mentioned later on, but here we're discussing it because it's directly related to volumes obtained from samples. The actual volume obtained from samples should be entered into LDMS, and a great way of doing this is to take the same tubes that are used in your site for sample collections and fill them with known quantities of water, and you can use these as measurement comparison tools. On the left are different examples of collection containers as provided from the manufacturer, and each of them have a slightly different volume requirement to consider that sample complete collection and usable sample. When we're discussing knowing the volumes that are being collected, we're not specifying that we want to know 18.62 milliliters of a sample were collected, but we are concerned with knowing that 14 mLs was collected per sample versus 16 mLs. The only way to get a specific volume would to be take the sample out and measure it completely. So the comparison tubes are really a great guideline that we suggest. And aliquot matching to containers is important. Here, somebody is matching a secondary container with the aliquot containers that the sample will be placed into for long-term storage. And this helps us, again, maintain integrity and the sample chain of custody. All HPTN samples are generally stored at -80 C until, unless they are otherwise indicated in protocol documents. There is a small range of acceptable temperature fluctuation for a long-term storage. We get a lot of questions about using a -70 system for storage, but these are not adequate because of their temperature fluctuations. And the HPTN does not utilize a central biorepository. All sites that participate in a protocol should be prepared to store and hold all the study samples until directed to ship or told that samples can be discarded. The LC requires that a group of site staff meet to review sample data across all systems that they have in place. So when the clinic documents their collections and enters them into a CRF, and tracking sheets are paired with tubes as they're transported, and the laboratory enters LDMS data, all of these different data points and systems should be discussed for visits so that there's uniformity of data entry and accuracy of those entries can be made. And here's just an example of a suggested storage system. and a storage system inside of the -80 freezers. Also, during a study, the LC is available to help with all the various reports that are distributed and requested. We will perform ongoing QA testing as dictated in each protocol or SSP. And we are available to provide on-site support for all labs and testing mechanisms related to activities whenever a site requests. QA testing is performed at the LC. This is dependent on the manual of operations, each protocol, and each SSP. LDMS must be used when shipping samples. There should be a manifest and a batch, either physically sent with the samples or electronically to us over email. When there's a clinic audit, one of the items that is checked is the physical storage against the LDMS data entry. The LC will help with reports after clinic audits, and any resolution of findings that require action on the site's part. At the end of a study and after a study, site staff continue to work with the laboratory center. We ensure all required and newly requested testing is completed before samples are ever discarded. The LC is happy to help with a number of laboratory activities, including EQA, any instrument or method validations, any audits or reports or action items that are ongoing. We will audit and review with sites GCLP standards to ensure that they're being met or the best way that a site can meet those requirements. And new this year are 2 evaluation criteria that the LC provide when network evaluations are reviewed. The first monitor is the quality of specimens and shipments, and this involves looking at the number of shipments that were requested and received within a specified time frame. And the requirement is that 90% of the requested shipments should be received within their specified time frame, and the LC tracks this information. The second monitor is specimen storage and completeness, and this involves knowing the number of aliquots that should have been stored, again based on volumes entered into LDMS, and checking this against the number of aliquots that are shown as available, and what the specimens were, what the visit was, and what the protocol is. And sites should have a goal of reaching 95% completeness for the storage requirement. And this data comes from an SDMC report provided to the LC for us to review and interpret. So in summary, the LC is always thinking about quality and integrity of the laboratory components of protocols. We are always available for discussion and help with laboratory and protocol questions. And thank you to everybody who helps us.
Thank you, Philip. So Dr. Emmel has been busy in the chat monitor answering a lot of questions. One, Dr. Emmel, that I'd like for you that I think would be useful for the group to hear, if you could answer, is what is the difference between the CASI and the Medidata Rave?
Yes, that's a good question. So Medidata Rave is used to collect the case report form data, or you can think of it in a clinical study as the clinical data. It collects sort of the basic information for the study, like demographics, enrollment, randomization, follow-up visits, adverse experiences, lab data, HIV test data. So all of those data go into Rave, and they're entered by the data manager at the site into the system. Cassie, on the other hand, is survey software. We use a software package called DATStat Illum, and it is meant for the participant to enter information confidentially about behavior into a separate data management. It's not data management, but survey software. So, and it is also web-based, like Rave, but you, at the site, you would have the participants sit down in front of the computer in a separate room and enter information about, a lot of times it's used for sexual behavior data or other sensitive information that the site staff wouldn't see. It is different also in that the software can be downloaded onto a computer and used in locations where there may not be internet access. And it's actually the software we've been using for the HPTN 071 pop art study to collect information as interviewers go actually into participants' homes, and the interviewer then asks questions that are displayed on the screen and enter the answers in a location where there probably is no internet. So the Rave is meant to collect the sort of basic study data, and the Cassie questionnaires are to collect confidential behavioral data most of the time.
And can you speak just a little bit about how confidentiality and then the security of the data compare between those two systems? You talked about it just a little bit during your explanation, but could you expound?
Well, they're both they both meet all regulations that the FDA, the U.S. FDA, and NIH require of data systems, and so all of the data is encrypted, meaning that it isn't easy to hack into and to see. So anyway, they both systems meet those requirements for electronic data. Did that answer your question?
I believe it did.
Okay.
The next question is for you, Phil. Is there a lab processing chart that details the amount of blood samples that need to be drawn for studies that are conducted by the HPTN?
So the HPTN LLC does not provide lab processing charts. Sample types and volumes are usually listed in the SSP or a site's chain of custody, and that is highly dependent on the site if it's in their chain of custody. The reason we don't provide a lab processing chart is because a CBC or an LFT volume and sample type is going to be very different in Thailand versus one of our U.S. sites. So we try not to be restrictive in that, because once we do put that in a protocol or an SSP, it becomes something that is very difficult for some sites to maneuver.
Thank you, Phil. Both of you touched a little bit on evaluation. Dr. Emmel, can you speak about how sites are evaluated as it relates to data collection?
Yeah, so we have sort of three sets of criteria that are used to evaluate sites from the statistical and data management perspective. One is data management quality, and so there are goals that we set for timeliness of data entry and the number of data queries, when there's an issue that either data that is inconsistent or missing, and submission of adverse experience data, those sorts of things. And then we also look at accrual of participants, enrollment of participants. Is it at the rate that is stated in the protocol or not? And then also retention of participants. Retention is a very important aspect of study management, and so the protocol specifies what sort of retention that we expect for a particular study, and so we evaluate whether the sites are retaining participants at that rate or not.
Thanks, Dr. Emmel. So we are out of time. If you do have additional questions, I want to encourage you. You can reach out to Dr. Emmel or Phil directly, or you can email me, Jonathan Lucas, JLUCAS@FHI360.org, and I can make sure that those questions are routed to the appropriate person. As a reminder, these slides, the presentation, will be posted on the HPTN's website, www.hptn.org. I'd really like to encourage you all also to complete the survey that's going to be sent out, the evaluation that's going to be sent out after this webinar. I really want to thank Dr. Emmel and Philip. You two are the best. Thank you so much for agreeing to host this particular call. Everyone, have a good morning and have a good day. Look forward to working with you all. Thank you.